afternoon, everybody. We are going to continue the afternoon session. Before we introduce you our plenary speaker, I would like to give you a few messages. First of all, I would like to remind you that this is the last opportunity to vote for the ecological carbon offsetting donation projects. Some of you may still be wondering what this orange card is all about. Well, if you don't know, remember this card is to vote for any of the 4 projects that have been chosen for the donation of this ecological carbon offsetting donation that we have into account in the Congress. Just as a very brief reminder, we have 4 candidate projects. 2 of them are oriented in the Amazon. One of them is the Madre de Dios Amazon project located in Peru. And the other one is the Yasuni Refuge of Amazonian Biodiversity, which is located in Ecuador. And then there are 2 other projects that are being promoted by the Vicente Ferrer Foundation. And one is about the wetlands to recycle wastewater. And the other one is about the effective use of water micro-irrigation systems. So please remember to cast your vote in the boxes that are in floor -1, because at the end of the closing session we will give the results of this vote and all the posters at the right time. Okay, so it is now my pleasure to introduce to the plenary speaker of this afternoon, Professor Jane Memmott, from the group of Ecological and Evolutionary Processes of the School of Biological Sciences of Bristol University. She has a very outstanding expertise and has provided very relevant contributions in many areas of research, such as food webs, pollination biology, or restoration ecology, among many others. And today she is going to talk us about the ecological restoration theory, practice, and reality. Please, Jane.
Hello. Right. First of all, I'd like to say thank you for the invitation to come here. It's an honour and a delight to be at such a conference in such a beautiful place. So, and term starts next week in Bristol, so this is kind of a final fling on the research front for a little bit. So, what I'm going to be talking about today is ecological restoration. And this is an outline of what I'll be going through over the next 40 minutes or so. So I'll start off with just a brief explanation of what got me interested in restoration ecology. And then I'll move through 3 main areas. We'll actually have a detailed look at what anthropogenic damage does to ecological communities. So I'm very much a food web person. I tend to look at everything in the community at once, if I can. So we're looking at the community scale, what's going on, and looking at 2 case studies. One, the impact of an alien plant on a heathland community, and then another one looking at how the productivity of a habitat is affected in a big restoration project running in the Azores. We'll then move on to the biggest restoration project I've been involved in to date, which is a 6-year study on British heathlands, looking at how we study the restoration and how we work out whether or not it's likely to have been successful, looking at restoration of pollinators, and then the things that eat the pollinators, the parasites, pathogens, and parasitoids that are feeding on the bumblebees. And then I'd like to end the talk by looking forward, what we need to do better, particularly if we're using this food web approach, to study restoration ecology. I'm going to argue, somewhat perversely perhaps, that we need both bigger networks and smaller networks for different reasons. We need a more theory, and in particular, we need better communication between academics, practitioners, and the public. So this was the paper that got me interested when I first started out as a lecturer in biological sciences in Bristol. I read this paper. This was a paper by Dan Simbaloff back in the 90s, and it's about restoration in New Zealand. And what he did was outline 3 problems or challenges, depending on your perspective, that were really affecting how restoration ecology was studied. First one he outlined as the fuzzy target problem. If we don't know exactly what a community is, how do we know when it's been restored? Second one, the moving target problem, and that's simply that communities are naturally dynamic. They're not static, and even in the absence of any sort of damage, they do change. And then the final one was an experimental design problem. And at that point, there was a real lack of scientific control and replication. And there were some real genuine problems here if you're dealing with landscape level restoration. And, you know, he kind of asked, well, what is the correct replicate or control for, say, the Isle of Guam? But I spent a lot of the last 10 years trying to address these problems, and it's been a huge amount of fun doing this, and I'm going to take you through some of the things I've been doing. So starting off on British Heathlands, a quarter of European heath is found in Britain, and it's the usual story with 80% has been lost. Actually, lost sounds a bit careless. It's been destroyed. It's mostly housing estates now, since the 1800s. And this is a habitat of huge biodiversity value and also cultural and historical value. And I'm going to take you through a study looking at what an alien plant has done to the kind of integrity of this particular ecological system. So one of the nice things about Heathlands is it's a very simple botanical community. You've got 6 or 7 common plant species, and that's it. That makes it nice from a restoration point of view, because you're trying to fix something that's relatively simple. Moving in here, though, we have some key alien species, and the one I want to talk about is one that's coming from the States. It's an ericaceous shrub called Gualtheria chelon. And this is the PhD student that did the work I'm going to tell you about. This is Luisa Carvalero. So what she did was she had 8 plots and a gradient of Gualtheria going from 0.1% to 80%. She sampled these plots, collecting all the herbivores in these areas. So you've got a mixture of heather beetles and heather beetle larvae and a whole bunch of leps. And then we did what we do in our group. We reared them out individually to put together a network of interactions between the plants, the herbivores, and the parasitoids. And I'm going to show you, this isn't one of her webs, I just want to show you how to read these webs. So we've got a 3-level food web here. At the bottom, we've got all the plants in the system. So each bar there represents a species. A wide bar tells you it's a common plant. So it's quantitative information. You've then got all the herbivores in the system, so all the caterpillars, and in this case there were leaf miners too. And then the parasitoids, the little wasps that feed on the caterpillars at the top. And then this tells you something about the frequency of interaction between the different trophic levels. So it's a snapshot of kind of the architecture of the community. And you can see that here we're starting to address some of these problems in terms of the fuzzy target. Well, we actually know we have a picture, we have a snapshot of what this community looks like. And it's not just homing in on 1 or 2 species, it's always looking at several hundred at once. And also the experimental design problem. If you replicate these networks, so if you have multiple ones, then you can do statistics, and then you can work out, you know, whether or not your restoration project is having a significant effect and so on. So we're starting to address the experimental design problem as well. So this is one of Louisa's webs. This is from a fairly uninvaded community. So we've got Gualtheria there, very, very low abundance. And as we go through the sorts of levels of invasion in her plots, you can see that as chelon increases, the structure of the food web changes. And that's just eyeballing it. If you look in detail, and Louisa did some industrial strength statistics on this, but in just a kind of a snapshot of what's going on, your insect abundance is decreasing, parasitoid species richness is decreasing as Gualtheria increases. You've got a disproportionate effect on the specialists. You've got disruptions of top-down regulatory mechanisms, changes in species evenness, abundance evenness, evenness of species interaction frequency. And the bottom line is, if you just look at the plants, you're seriously underestimating the impact of what's going on. The effect of this plant is rippling through this community. And restoration has a big job to do. So moving to a different system, looking at it from a different angle, but asking the same sort of question. So this is working in the Azores. So just so you can see where the Azores are, it's about a third the way between Portugal and the east coast of the States, and it's an archipelago of islands. And if you've not been, you ought to put it on your list of places to go in life, because it's a fantastic place to go as an ecologist. This is the island we're working on, the island of San Miguel. And if you look at the colour coding here, basically this island looks largely pink. So most of it has actually been invaded by alien plants. The grey stuff is the production forestry, so an alien tree, Cryptomeria, is grown commercially. And that one little green spot is the patch of native vegetation that's left on this particular island. And this is a story that's repeated around the world, on islands, you know, all over the place. In that little patch of green lives a rather special bird. So there's an endemic bullfinch that's found in this habitat. And we were kind of piggybacking on the back of a big EU-funded project looking at this particular species and restoring the habitat for it. So this is our headquarters, a particularly delightful town with very nice cake and coffee shops in it, down on one end of the island, kind of headquarters. So alien plants, as you all know, they're a very common problem on many, if not all, restoration projects. And the question we're asking here is how alien plants affect the productivity of a habitat. And we're looking particularly at productivity in terms of insect biomass. And this insect biomass feeds into the rest of the food chain. So if you're an insectivorous bird or lizard or spider or whatever, this is important currency to you. And this is a very different approach to looking at, you know, individual species. We're looking at a kind of community level response here. This is Ruben Helano, who did the project I'm about to tell you about. So just a bit of background to the project. In the back of the picture there, you've got native Azorean forest. Then in the front, you've got one of the key invaders. So this is one of the gingers, the Hedychiums, a plant I became very familiar with when I worked in Hawaii. And this is Jane Memmott, who's the Ruben's co-supervisor that did all the bird work on the project, and was a delight to work with. So what they're doing on this project is they're restoring a mountain range. And they have huge teams of people that go through armed with herbicides that spot treat the alien plants across a huge area. It's a tremendously impressive project. It's like warfare. It's a huge logistical operation. And that's the kind of satisfying picture at the end of it. You've got a very sick-looking ginger there. You can see the dead tubers, and then the natives coming back behind it. So they're basically weeding a mountain range. Going back to the question, we're asking about how alien plants, which is a mixture of the gingers, and there's 2 or 3 other key plants in this system, affect the productivity of this habitat. What Ruben did was actually sample a number of plots, 35 plots, with varying amounts of the alien plant. So a similar approach to Louisa. And as you can see looking at this picture, this is what I call extreme sampling. Okay, it's a hard habitat in which to work. This is the experimental design. So we've got these plots scattered over the mountain range. They're sampled 3 times a year. Oh, we have a technical hitch. So someone going to rescue me here. It's frozen. There we go, we're there. Right, so recovering swiftly. This is the network, all of the data put together for the 3 years of the project. So this, I'm going to take you through this network. In the middle, you've got the seeds. So we're looking at, rather than looking at leaves or flowers, we're looking at the seeds in the community. So we've got all the plant species across the middle there. We've got, again, a wide bar, it means it's a common seed species. A narrow bar, it's a rare one. Going down the food row, we've got all the birds in the system that feed on the seeds. We've got the mites that feed on the birds that feed on the seeds. We've got all the herbivores that come out of the seeds, so the flies and weevils and moths and so on that are feeding on this mixture of native and alien plants. And then finally, we've got the parasitoids that are feeding on the herbivores. And then just to remind you, that's Ruben that put that huge amount of data together. So looking at what we found out about this particular system, and that's actually that's the endemic bullfinch. You can see that's just a tiny, tiny part of this overall system. And And these are the alien plants shown in orange. So those are the ones that have been removed during this habitat restoration. So we made two predictions, and these are kind of almost, they're very straightforward predictions that me as a simple field biologist, you can almost do these on the back of an envelope, but they give you some really useful and really interesting ballpark figures. So we know the biomass of all the seed herbivores in the system because we can simply weigh the things that emerge from the seeds. And we're going to predict the changes in biomass across this mountain range in the worst case scenario and the best case scenario. So the two extremes. We're going to ask what happens if alien plants take over the mountain range completely. Complete ecological disaster. And we're also going to ask what happens if we manage to successfully remove all the alien plants and restore the natives to the community. Let's see if we can get that little... damn. Hang on, I was trying to get rid of that box then. Which one is it here? So we're going to... hang on. Oh yeah, thank you. Right. If alien plants outcompete the native plants, what we're looking at from this calculation is a 67% decrease in insect biomass. So if you're a seed-feeding bird or a spider, that's not good news. Looking at what happens if alien plants are successfully removed, though, by the restoration program, we're looking at doubling current biomass. So as I say, these sorts of ballpark figures give you some idea about what's happening across the whole community. So moving on to actually assessing what's happening to the community once it's been invaded, we're going to look and ask, can we actually restore structure and function to these communities? And this is looking at a big heathland restoration project in the UK. So these are the two PhD students that did this work, along with my trusty hound. And it's the first project I had looking at the restoration of ecological interactions. So this is just moving into restoration ecology. So Michael Forrett was a Danish PhD student, and he did three things. He put together plant pollinator networks. He sampled them twice, 11 years and 14 years after the restoration had taken place. And this is restoration from forestry. So these heathlands have been heathlands for 3,000 years. Commercial forestry came in, and then commercial forestry went out of favour, and they were restored back to heathlands. And the experimental design was we had four pairs of webs. This is a kind of cartoon of the system. So you've got a simple plant community and all the flower visitors that come in. So not just the bees looking at the flies, the weevils, everything that visits those flowers. And again, we're starting to see we've managed to get into all three of these challenges. We've got the fuzzy target problem dealt with in that we've got these snapshots of the networks. We've got data twice, 11 years and 14 years after the restoration. And we've got a paired design. So we're starting to kind of be able to do statistics on things. We're asking three questions. Asking whether pollination's been restored. Asking whether the restoration is robust to future changes, because we don't want to be kind of gardening long term. We want these systems, these restored systems, to be sustainable. I mean, you look specifically at the effect of future species loss on these communities. And then the question Kate addressed was whether bumblebee parasitoids, parasites and pathogens have been restored. And in the past, I think there's been a tendency to look at the kind of iconic things in communities. And, you ask a restoration, a conservation management, do they want parasites and pathogens back? And they used to say, well, no, absolutely not. But these are a key part, they're an essential part of a healthy, intact ecosystem. And in some ways, they're the last things to move in, so they make quite good arbiters of success. So what we do in these, just like Louisa's project, you've got a plot, you run transect lines, and you're looking at the interactions between the pollinators and the flowers. So there we've got 3 bits of data in that photo. We've got a Latin binomial for a bee, a Latin binomial for a plant, and the interaction between the two. There's then a large taxonomic phase. I have very good links with taxonomists, and so all of our pollinators are done by taxonomists at the National Museum of Wales, because you can't do this sort of project without taxonomic help. They're absolutely key to our success in trying to put these networks together. So they're identified, they come back, but before they're sent off, they're swabbed for pollen. and we identified all the pollen grains, the thousands, tens of thousands of pollen grains on the insects. And the pollen grains are absolutely gorgeous. They're like alien spaceships when you look at them close. And by the time you look at about 3 million, you find one smiling back at you. So it's actually great plantain, that, and it's just a 1 in a million pollen grain. But it made us all laugh at the end of a very long day. Right. We use the information on visitation to put together networks that describe the quantity of visitation, who visits whom and how often. So just quickly taking you through this, at the bottom there you've got the flowers. So the two dominant plants will be Calluna and Erica, and then you've got some gorse and a couple of other things in there. You've got all the flower visitors at the top, colour-coded. In red it means they're Hymenoptera, blue means they're Diptera, and then you've got a couple of leps and a couple of beetles probably in there. And then again, just like the parasitoid and caterpillar data, you've got the frequency of interaction. So that's the quantity of visitation. We also have data on the quality of visitation by looking at the abundance and diversity of pollen loads on these insects. We can combine these two bits of information in a way I'm going to gloss over a bit now, but basically you can come up with a pollinator index that ranks the visitors as likely pollinators in your habitat. So returning to our first question, we're asking whether pollination is restored or not. Well, in total across our 8 sites, we had 80 things visiting, 80 species of insect visiting the flowers. There were far fewer pollinators, and we can tell the pollinators, well, we can tell the cheaters because they have no pollen on them. We still can't prove pollination, but if they're carrying the pollen and visiting the flower, we're starting to get some really fairly substantial evidence that they are pollinating. And we found there was no real difference, no difference at all, no significant difference between the ancient and restored site with respect to the key pollinators at each site. So there were the apis, the bumblebee species, and also a species of hoverfly, Episurfus balteatus, was an important one in this particular system. So looking at the resilience of these habitats to future species loss, because things will happen in the future that will potentially lead to species loss, we found a trend in the ancient sites were more resilient. And what we're doing here, just so you can know what's happening, is you're taking the webs in silico on the computer and killing off species one by one and asking at what point the community collapses, and is there a difference between the ancient and the restored sites. We do have a problem with this. This is the first restoration project I ran, and our statistical power is actually quite low because we only have 4 pairs. So one of the things we learned from this project rather quickly was we need a bigger sample size. But it was the first time people had actually thought about asking about the resilience of restoration projects. And it led on to much bigger projects where we have things like 10 pairs that allow us to test resilience much more accurately. So we've got a trend, but whether anything's actually happening or not, we don't know. But it was a very interesting question to ask at the time. Then moving on to Kate Henson's work, what she did was she collected just the bumblebees. They go straight into the freezer, and then she spends the winter dissecting each bee. And I remember saying that when you actually dissect these bees, it's a bit like a Christmas present. You never quite know what's going to be inside these things. And there's actually 9 different things that lived inside our bees, from these giant, I mean, they're not as giant as you see up there, but these are blowfly-sized larvae that are living inside. They fill the whole abdomen of the bee, so canopid larvae. Over on the far side, you've got tracheal mites, so little tiny mites that live inside the tracheae of the bee. You've got some worms, and then the little protozoans down here are rather fun because the bees have spent a few months in a -30 freezer, and they're obviously absolutely 100% dead. But when you take them out and thaw them, some of the protozoans come back to life, and you can see their flagellae kind of wriggling and things, so they're very easy to spot. But there's something slightly spooky about something that can cope with being at -30 for a while. So she dissected all the bees and used this information to put together networks that describe the flowers, the bees, and then the things living on the bees. And down the bottom there, you can actually see what the real networks look like. And what she found was that we'd actually gone up to 12 sites at this point. We've got 6 pairs of sites rather than 4 in an effort to get more statistical power. She got 9 species of bumblebee, 9 species of parasite, pathogen, and parasitoid, and 8 of the 9 of these things were restored. There was one missing, and it's the canopid flies are still missing. 14 years after the restoration, the plants are back, the pollinator communities are back, but this last little bit of the jigsaw isn't back yet. And that's really quite interesting, because people don't generally notice things like canopied flies, but they're really quite rare in their own right. There's only about a dozen species in the UK. And all, the emphasis on heathlands before has been on things like some of the more attractive butterflies, dark-footed warblers, smooth snakes, and so on. So these little less charismatic things, if you look for them, you can learn some interesting things. So, moving forward, looking at what we need to do better as restoration ecologists to be able to fix these damaged and degraded systems more efficiently and effectively. So I'm going to start off by asking about what can we do with these networks to make them more useful and to provide better information. And I'm going to argue that in some circumstances we need to sample more, and in other circumstances sample less. And something that had been niggling me for about 5-6 years ago, I was very much aware that different parts of the literature have got different types of networks in. So you've got pollination networks in some parts of the literature, parasitoids in others, seed dispersal networks, predation networks. And this is kind of explained by the taxonomic comfort zone of us ecologists. So you've got pollination ecologists working on the bees and hoverflies, applied ecologists working on the parasitoids, ornithologists working on the seed dispersal. And I'm absolutely as guilty of this as anyone else. We work on what we know. But in reality, in any field or wood or heathland, these networks are linked. They're not independent. Colin's done a great job at providing a general perspective on this, and I was delighted to be involved in that project because I'd been trying for the previous 3 years to actually put together a network of networks. And I want to tell you about this project because this is to do with making networks bigger and what we can learn from them. So this network, which we call the Norwood Farm Network, is dated from a single large organic farm in Bath. It describes what's going on in a community of 204 plant species, 690 invertebrate species, 75 birds, 19 mammals. We sampled so hard and so long, we found a new species of parasitoid to the UK. But you can see from the diversity of pictures there, the range of organisms we're sampling. And the range of techniques we used. This is a seriously non-trivial task. Trying to put foxes and flies into the same network, measured in the same way, is actually really a bit of a challenge. So here we're sampling those aphids and pollinators. There's me up there in the top corner having fun on a seriously cold and unpleasant day, emptying the rodent traps. We're catching birds, rodents, insects, you name it, we counted it. And reared a lot of stuff as well. And these two people down the bottom here, Michael Pocock and Darren Evans, are the two postdocs that led this project. And this is kind of a schema, an outline of what we were trying to do. We were trying to produce a network that had people in it. It had anthropogenic things in it. So it had crops and livestock in there. A network that encapsulated more than one ecosystem service. So it had pollination, pest control, and seed dispersal in there all at the same time. It had the bioindicators in there. So, where are they? Do they show us anything useful? And it also had a vast swathe of what we kind of call hidden biodiversity, all those grotty little dipterans and little brown beetles that people tend to ignore. We wanted to get as many of those in as possible. And you can see from this cartoon, we've got at the bottom, we're sampling all the parts of a plant. So we've got the leaves, the flowers, and the seeds. We've got the birds, we've got the mammals, we've got a whole bunch of insects. We've got some of the parasites on the mammals, so a great deal of fun we'll have catching rodents, and then you comb them with a toothbrush and get the fleas off them. So we did all sorts of strange and interesting things on this project. And this is what the overall network looks like. Now, it's actually quantitative data, as you see at the moment, but it's very difficult to display in a quantitative way. Down the middle, there in green, are all the plants. And then these are all the individual networks that fill it out. So we've got various pollination networks, we've got an aphid network, a leaf miner network, seed feeder networks, and then ones with rodents and granivorous birds and the fleas on the rodents and so on. And these are all sampled in the same place at the same time and on the same sort of scale. The sorts of things we're starting to ask with this networks, this is work that's in progress, is we can start to look at the robustness of the individual networks, asking, are there certain types of networks that are much more fragile than others? Because if there are, then there should be targets for restoration projects. They're the ones that need fixing first, or fixing fastest. We can also start to identify the most important plants in these systems for other species. Because the ones that actually have a disproportionate effect, these are the kind of targets for restoration in agro-environmental schemes. And rather interestingly, it turns out the most important plants are the perennial weeds, which is going to be an interesting one when trying to sell this idea to farmers. But at least they have the advantage that they're easy to grow. So that's one way of doing things, making webs bigger and better so you can get more and more information from them. But the opposite argument for that is if food webs are going to be used by practitioners, so that people in the field practically trying to do conservation, they actually need to be quicker and cheaper to make. So Norwood Farm was big and expensive to make. Basically in conservation, they rarely have excess cash. Time is money. So we're looking for ways to actually cut corners when making networks, making quick and dirty networks that will allow us to quickly get the information we need. So the question is here, where can you actually cut the corners? Well, we got a first inkling of this on the Hawaii project that ran back about 10 years ago, run by Laurie Henneman. And there we were looking at where alien parasitoids released for biocontrol projects were getting in natural communities. And to do that, we produced 4 networks in a really rather challenging habitat. And we did that. And then a couple of years later, we thought, well, did we really need to kind of sample 2,000 caterpillars? If we resample the networks in silico and ask the same questions, how little sampling could we have got away with and still picked up the main patterns? And it turned out if we'd have done 10% of the sampling, which is an awful lot of days less in the field trying to catch these things, we could have got the same sorts of answers. We could have reared 200 caterpillars rather than 2,000. Now, we probably wouldn't have got that past the referees, but it's an interesting idea that you can sample a lot less. But exactly how do you sample less? Where do you cut these corners? Well, I had a really fun go at answering this question with a Norwegian visitor to the lab that came over for 6 months. So Stein Hegeland arrived from Norway with a fantastic pollination data set, gathered from some gorgeous alpine meadow in Norway, from a single site over 2 years. And what we did with this network was to resample it in various, basically doing less and less sampling per time to work out when we started to lose information. So these are the sorts of ways we sample the data set. We started off with our full data set, which is 2 years worth of work, 22 weeks of sampling, 132 transects. So this is 2 years of a PhD student's life, basically. And that's far too expensive for conservation ecology. We then looked at just 1 year's data, so the 2003 data and the 2004, so we halved the sampling. We resampled it just once a week. We only sampled 1 sample out of 3 transects. Then getting kind of quicker and dirtier, we just sampled the peak field season. So it's just a month sampling rather than 2 years, 22 weeks of sampling. And then our most minimalist sample was just doing 3 transects across the whole season. And just to give you some idea of the sorts of results that came out of this. If you just look at the peak season from one of the years, so July 2004, we could actually identify 85% of the functionally most important pollinators. But in terms of cost, in terms of staff time and travel time and taxonomy time, so all of this was costed out, it actually worked out at 20% of the full cost of a season. So you can cut corners. It also provided good estimates of the main network descriptors. Now, the catch in this is you actually need to make the big network once. You still need the big network to start with. But if you resample in silico, it allows you to ask how little sampling you can get away with in future studies. So if, for example, in that Norwegian system, we wanted to ask about, say, meadow restoration across Norway and wanted to sample 20 sites, we know the best way to work out how little sampling we would have to do. We also need better theory, and I want to just tell you a little bit about something we're doing on a restoration project working in Caledonian pine forests. These are the ancient forests in Scotland. They originally covered 70 to 80% of Scotland, and only 1 to 2% of them remain. They're far more endangered than tropical rainforests. This is one on a nice day in the summer with Mariano Devoto, who worked on this system. Came over from Argentina to work with me for 3 years. And just as a little aside, I sometimes think I make these networks look too easy to make. And so just to show you how challenging they can be in the field, Mariano had a particularly tricky time with his networks. And one of the things he had to deal with was the Scottish weather. And I mean, Scotland has, this isn't quite Scotland in summer, but it felt like it some of the time. And at the end of his first field season coincided with the worst summer in Scotland for 100 years. Okay, he was sampling 30 field sites from April through to September. At the end of his first field season, he had 123 insects. From these 30 sites, it was a deeply unhappy Mariano at the end of that first field season. But the following year, the weather came out, the sun shone a little bit, we changed our sampling protocols, and we did much better. We got a couple of thousand pollinators. You can tell the body language, it's just great. So, these networks are, the amount of blood, sweat and tears that goes into these networks shouldn't be underestimated. And I'm incredibly lucky to work with such fantastic people that do this work. So this is the data we ended up with after that second field season. And you can see how our approach has changed over the years. We've got 6 replicates of 5 different stages of a managed succession. We've got 30 networks. You can start to do all sorts of interesting things when you've got this much data. And we've got data on a managed succession through young plantation, through various stages, along with ancient forests in these few remaining fragments. And what we're starting to do on the theory front is actually modelling the system in silico, but looking at restoration in silico. But we're using the reverse of the methods used by Jennifer Dunn and by myself and various other people, looking at extinction cascades. Because rather than killing species off one by one, we're putting them back in one by one, according to various restoration scenarios. So if we want to aim to maximise robustness, we can do that. And this is leading to some really, really rather fun results. The other thing we're doing on the theory front is I'm actually a simple field biologist, but I think good theory is incredibly important. I think it was Einstein that said that there's nothing so practical as good theory. So even though I'm not a theoretician, I'm very, very keen to get more theory into restoration ecology. And one thing that's been particularly fun to do is I have a theoretician in my lab at the moment called Daniel Montoya. And we noticed to introduce theoreticians to practitioners is really rather fun because I interact with the practitioners, but theoreticians and practitioners don't tend to meet in their normal kind of everyday life. So actually getting the 3 of us together, a field biologist, a practitioner and a theoretician in the same room to kind of work out what are the big questions that the practitioners need answers to that are also interesting on the theory front is really rather fun. And then just to end, I want to talk about better communication. Now, one thing that became fairly obvious fairly quickly is if you want to influence policy, it is really, really hard. We start off writing into grants that these results will have implications for policy, but to actually get an effect on policy is difficult. But you can influence local restoration issues by working with your local practitioners. It's simple and it's a huge amount of fun, and I think it's really beneficial to science and to the practicalities of restoration. And I want to tell you briefly about 2 things I've done, and these are 2 things that anybody can do. So just to define what I mean by practitioner, and this is a definition from an editorial in Nature a couple of years ago. So they're defined as these are the people at the coal face. These are the people who buy the land, who put up fences, set fires, put out fires, lobby politicians, negotiate with farmers, spray invasive weeds, poison rats, and guard against poachers. These are the people that manage the nature reserves. First thing I did was organise, this is 3 or 4 years ago, and I'd just become an editor at the journal Applied Ecology. To organise a 1-day local conference to get academics and practitioners talking together on my own patch, which is Bristol. So it's a big city in the west of England. So what we did was we invited all the practitioners we could find that worked in the city of Bristol. And these come from the wildlife charities, government scientists, soil association, and so on, all sorts of different walks of life. Got together the ecologists in my own department and ended up with a conference of about 70 people, a mixture of academics, PhD students, and practitioners. And then we had a 1-day symposium of 10 talks, 5 academics, 5 practitioners, and I alternated them one by one, so it wasn't kind of like a his and hers type thing. And they're talking in pairs on the same general topic. I added lots of cake and coffee. I'm a great believer in that chocolate biscuits make the world go round. And there was a real buzz of conversation. Lots and lots of interactions between PhD students are brilliant. They're the future of ecology. And getting them and the practitioners talking together was just wonderful. And several things did come out of this 1-day meeting. It was a lot of fun to do, and I'd like to lay down a challenge here. Try doing one of these. Find out who the practitioners are, close, within a 20-mile radius of your university, and invite them in for a conference. Coffee and cakes are important. Otherwise, it just works. It's a huge amount of fun, and as I say, it led to all sorts of things happening after this, just by talking. The other thing I've done, and this is at the moment the big project in the group is a big urban ecology project. So I'm leading a consortium of 28 people in all through some very, very interesting questions on urban networks. Before I wrote the grant, I made contact with the practitioners I wanted involved in them. It's really important if you want to influence any sort of, anything that's going to happen in the field, to talk to practitioners and get them involved in the grant before you write it and submit it, and write them in as collaborators. Because to tell them about your results after you've done it, it's not as effective, they're not as engaged with it, whereas if they're involved in the planning, the doing, they can see the results coming through, I think there's a far greater chance of success. So this project had written into it, there's 5 of us academics employing 5 project staff, 7 practitioners, so I've got the county ecologist in 4 cities written into this grant, along with the head of reserves of the Wildlife Trust. They go in on the forms, they see the grant before it goes in, and there's also 3 taxonomists written in. So they're involved all the way through the project, and you can see them all there. Everyone but the dog is a member of this consortium. And then, so they go all the way through the project, but then at the end, I wrote into the grant funding to completely cover the cost of inviting 110 practitioners to a conference in Bristol at the end. And again, conservation and restoration is beset with a lack of money, but if I can cover their travel costs and their accommodation costs, so we can tell them what we found rather than expecting them to find it in the scientific literature, I think we're more likely to actually be able to do some of the things we say will work in urban habitats. So I think impact is much more likely. And it's also a huge amount of fun doing this, working with practitioners in this way. So I'd highly recommend it. So, in summary, I've talked something about damage to habitats, the way plants can actually cascade through whole systems, looking at restoration ecology in a heathland context, and some ideas for improving how food webs can actually work, more communication between theoreticians and practitioners. And I've also laid down a challenge to actually run one of these one-day conferences sometime in the next year or so. And finally, just thanks to these are the people behind putting together these networks. So it's a real pleasure and a privilege to work with this bunch of people. These are the ones that actually are out there gathering up all those interactions I've shown you today. And thank you for your time and attention.